Unlike myoglobin, most eukaryotic proteins are composed of two or more spatially distinct modules or domains that fold independent of one another. For example, the mammalian enzyme phospholipase C consists of four distinct domains colored differently. The different domains colored differently in case of drawings. The different domains of a polypeptide often represent pairs that function in a semi-independent manner. For example, they might bind to different factors such as coenzymes and a substrate or a DNA strand and another protein, or they might move relatively independent of one another. Protein domains are often identified with a specific function. For example, proteins containing a pH domain. Protein domains are often identified with a specific function. For example, proteins containing a pH domain bind to membranes containing a specific phospholipid, whereas proteins containing a chondroitin bind to a methylated lysine residue in another protein. The functions of a newly identified protein can usually be predicted by the domains of which it is made. Many polypeptides containing more than one domain are thought to have arisen during evolution by the fusion of genes that encoded different ancestral proteins, with each domain representing a part that was once a separated molecule. Each domain of the mammalian phospholipase C molecule, for example, has been identified as a homologous unit in another protein. Some domains have been found only in one of a few proteins. Other domains have been shuffled widely about during evolution, appearing in a variety of proteins whose other regions show little or no evidence of an evolutionary relationship. Shuffling of domains creates proteins with unique combinations of activities. On average, mammalian proteins tend to be larger and contain more domains than proteins of less complex organisms such as fruit flies and yeast. Polypeptide chains greater than of 200 amino acids that fold into two or more compact globular clusters are called domains. There are three main types of domains. Alpha domains are composed of alpha helices. Beta domains contain antiparallel beta sheets and usually contain two beta sheets packed against each other. Alpha by beta domains contain beta alpha beta motif of parallel beta sheets. Adjacent domains are connected by one or two segments of the polypeptide chain. All globular proteins have a defined structure. In the tertiary structure of protein, almost all the hydrophobic side chains are found in the interior of the protein, and almost all hydrophobic side chains are found on the outside of the protein, interacting with water. Globular proteins are compact and there is no space inside, hence, water is effectively excluded from the hydrophobic interior. Nearly all hydrogen bond donor example studying for hydrogen bonds with hydrogen bond accepts like glycine. Hydrogen bond formation neutralizes the polarity of the hydrogen bonding group. Beta sheets are usually twisted or wrapped into barrel structures and loops and turns around the unit side outside of the protein. Mutations which place a hydrophobic side chain on the surface can cause significant change in the folding of the protein.